Estamos ao vivo.
Oba!
Boa tarde a todos e todas. É com grande satisfação que a gente inicia o nosso 5º evento do programa de pós-graduação em Biosciências. É uma grande satisfação estar aqui com vocês, nesse momento tão crítico de pandemia, mas que as ferramentas virtuais elas têm essa capacidade de nos aproximar. Então, para quem não me conhece, eu sou a professora doutora Elisandra Braganhol, e atualmente eu coordeno o programa de pós-graduação em Biosciências. E esse nosso evento, então, é o 5º Bioscience Meeting Crossing Borders. Nós iniciamos, então, saudando a todos os presentes nessa sala virtual e aos que nos acompanham pelo canal do YouTube, no nosso programa, coordenadores de curso de graduação e de pós-graduação, alunos, professores, servidores e a comunidade em geral. Eu cumprimento de forma muito especial a nossa vice-reitora, orientadora e a fundadora do nosso programa de pós-graduação em Biosciências, a professora doutora Jennifer Saffi. Então, para dar as boas-vindas, eu passo a palavra, então, à nossa Jennifer, né, para fazer a sua palavra aqui, né. Estamos muito felizes com a tua presença aqui, Jennifer.
Eu é que estou, Elis, muito obrigada. Então, boa tarde, colegas, estudantes, pesquisadores que nos assistem, né, pelo YouTube. Quero fazer uma saudação especial aos nossos palestrantes também externos, a nossa pró-reitora de pós-graduação, professora Márcia Jovenardi, que também é professora deste programa, professora Elis, né, coordenadora do PPG Bio, em nome de quem eu saúdo a todo o nosso corpo docente aqui do PPG Bio e também da comissão organizadora do evento. Eu fico muito honrada em representar a nossa reitora, a professora Lúcia Pelanda, nesse evento, porque eu não falo apenas na condição de vice-reitora, mas como também a Elis já citou, eu sou professora, né, integrante desse PPG, que eu ajudei a constituir lá em 2014 ainda, né, junto com um grupo muito comprometido e motivado de colegas. E todos nós tínhamos a convicção que a nossa universidade já tinha uma massa crítica e já tinha uma infraestrutura, né, que suportavam a criação de um programa na área de ciências biológicas em nível de mestrado e doutorado, porque até então todos os nossos programas eram em outras áreas da CAPES. E eu tenho o maior orgulho de acompanhar o crescimento e a consolidação das linhas de pesquisa e da excelência na formação de mestres e doutores do CPPG ao longo desses últimos 5 anos, né? Já estamos com 5 anos de PPG Bio. Eventos como esse, né, que também foi pensado já no momento da estruturação do PPG, eu considero fundamental para que a gente possa refletir e discutir ciência e pós-graduação de uma forma integrada, contando com a experiência e a competência de pesquisadores do Brasil e do mundo. E o tema escolhido, né, para essa edição, que é Cruzando Fronteiras, não poderia ser mais apropriado, ainda mais iniciando no dia de hoje, que por si só também é um marco para a ciência mundial, pois é o dia em que oficialmente, então, é dada a primeira vacina, né, contra o Covid-19. E esse ano super atípico, ele foi muito desafiador para todos nós, em vários sentidos, né, do ponto de vista pessoal, também profissional, e para a nossa universidade é importante dizer que ele é um ano de muito significado, né? A UFSPA, como a única universidade federal brasileira especializada em saúde, desde o início desse ano, né, do início da pandemia, ela teve um papel muito ativo no enfrentamento, né, do Covid-19. Não só a universidade não parou, mas ela também intensificou as suas atividades. E esse grupo que está aqui hoje participando do evento teve e tem um papel muito relevante nesse protagonismo, ao representar o nome da nossa universidade em vários cantos do Brasil e do mundo, dando entrevistas a diferentes jornais e TVs, dando palestras, escrevendo, aprovando projetos de pesquisa de extrema relevância. E essa é a maior prova, né, de que em momentos de crise, que as universidades, com o seu grupo de profissionais e pesquisadores, é que devem assumir o seu protagonismo. É sempre bom lembrar que mais de 90% da pesquisa no nosso país é realizada nas universidades públicas, que geram conhecimento para solucionar problemas a partir de demandas da sociedade. E esse ano foi a maior prova disso. E Sem investimento em educação, ciência e tecnologia, jamais chegaremos à condição de um país desenvolvido e soberano no enfrentamento dos nossos problemas, das nossas desigualdades e dos nossos contrastes, que ainda são muitos, muito grandes. Nós podemos estar enfrentando a maior crise da nossa história, mas enquanto for possível, nós cumpriremos a nossa missão, buscando melhorar a cada dia, buscando contribuir para uma sociedade com maior qualidade de vida, que seja mais justa e, principalmente, né, mais humana. E momentos como deste evento, também servem para reafirmar o nosso compromisso com a sociedade, nos fortalecendo em defesa, né, de ciência e da educação no país. Então, Elis e colegas, eu quero parabenizar mais uma vez a todos que se engajaram na organização deste encontro e principalmente também parabenizar os nossos alunos do PPG Bio pelos vídeos maravilhosos na divulgação dos seus trabalhos. Eu agradeço de coração também os nossos palestrantes dos diferentes cantos do país e do exterior. Eu acho que o formato virtual também nos permite ficarmos mais próximos, né? Cruzando essas fronteiras com o mundo. Eu quero deixar também um abraço muito especial da professora Lúcia, que também elogia demais os vídeos dos nossos alunos. Fica o meu grande desejo e a certeza, né, de que esse evento será de muito aprendizado e de muito sucesso. Muito obrigada e um ótimo encontro para todos nós.
Obrigada, Jennifer, pelas palavras. Eu pego o gancho da questão da atividade dos vídeos de divulgação científica. Essa foi uma ideia dos alunos que fazem parte da comissão organizadora do nosso evento. E eu, enquanto coordenadora do PPG Bio, eu sou muito grata ao nosso grupo de professores e ao nosso grupo de alunos. Eu percebo, assim, uma sinergia imensa e é o trabalho em colaboração, é o trabalho em sinergia que faz a gente alcançar os níveis de excelência e é o que faz a gente conseguir chegar mais longe. Então, eu gostaria de deixar registrado que foi uma iniciativa deles e eu achei sensacional e a gente abraçou a ideia e propomos atividade e todos eles, né, o resultado está aí, foi surpreendente, né?
Maravilhoso, parabéns mesmo.
E deixa eu pegar, eu preparei algumas palavras para falar aqui para o pessoal, eu vou pegar minha colinha aqui, porque eu sabia que eu ia estar um pouquinho nervosa hoje, então eu resolvi deixar uma coisa escrita aqui para não perder as ideias que eu quero passar para vocês, né? E eu gostaria de continuar a minha fala, né, fazendo algumas considerações sobre a vocação do PPG Biosciências e da importância da manutenção do nosso evento anual, mesmo em um ano atípico de pandemia. Desde a sua origem, o PPG Biosciências tem o objetivo de congregar a comunidade científica para discutir ciência, pesquisa e inovação, com pesquisadores de referência em suas áreas, integrando professores e alunos de todos os níveis. Nas edições anteriores, nós nos dedicamos em abordar temas focados em genética, fisiologia, biologia celular, e o último encontro, então, nós focamos em desenvolver atividades focando nas 4 linhas de pesquisas desenvolvidas pelo nosso programa. Porém, esse ano está sendo diferente, todos nós estamos percebendo isso, né, pessoal? Sem sombra de dúvida, estamos vivendo um período histórico em que, mais do que nunca, a ciência e os cientistas assumiram o seu protagonismo no enfrentamento da pandemia. Particularmente hoje, dia 8 de dezembro, é um dia que todos nós iremos contar aos nossos descendentes. O dia em que iniciou a vacinação contra a Covid-19, apenas um ano após a pandemia ser deflagrada. É com esse espírito de valorização à ciência, aos cientistas, e contra qualquer tipo de pensamento obscurantista, que o evento do PPG Biosciências inicia. E esse ano vem propor um desafio, o Crossing Borders. Mas o que nós queremos dizer com esse Crossing Borders? Nós queremos dizer cruzar as fronteiras. Mas quais fronteiras? As fronteiras geográficas, as fronteiras do conhecimento, as fronteiras que separam o conhecimento científico da vida cotidiana das pessoas. Sim, é preciso que as pessoas compreendam que a ciência está em tudo que nos cerca, e que, por isso, deve ter seu valor reconhecido. Para isso, então, nós montamos uma programação repleta de atrações. Contaremos com a participação de cientistas de universidades canadenses, americanas e brasileiras. Nós já iniciamos as atividades do nosso meeting com os alunos fazendo divulgação científica para o público não especializado. E aqui eu gostaria de fazer um convite especial a quem ainda não está acompanhando o Instagram do PPG Biosciências, que não perca essa oportunidade, porque ali vocês vão encontrar informação muito relevante, informação de qualidade, informação complexa, apresentada de um jeito leve, que foi o objetivo da nossa atividade. E, por fim, nós vamos finalizar o nosso evento com uma conferência e com uma mesa redonda de discussão a respeito da importância da divulgação científica para a sociedade e da importância da construção de pontes entre universidades e a população em geral. Vou finalizando aqui minha fala, destacando o compromisso que o PPG Biosciências assume em formar recursos humanos em alto nível e com capacidade para atender os anseios da nossa sociedade. Nós sempre temos focado nisso, pessoal. Nós propomos atividades, propomos esse evento particularmente, e o nosso objetivo de tudo isso é desafiar os nossos alunos, é desafiar a nossa comunidade científica para que a gente consiga extrair o melhor, para que a gente consiga lapidar essas joias que nós recebemos, e nós, enquanto orientadores, nós temos o dever e o privilégio de fazer essa lapidação e de entregar um profissional de qualidade para a nossa sociedade. Eu gostaria também de deixar expresso aqui o meu agradecimento aos palestrantes, que gentilmente aceitaram o convite para participar do nosso evento, aos professores que ministrarão os minicursos. Ontem nós já tivemos uma atividade sobre um curso sobre como utilizar a ferramenta R, que já foi sensacional, já começamos com o pé direito o nosso pre-meeting. E eu gostaria de agradecer também os avaliadores científicos e também os avaliadores artísticos. Nós temos, sim, 2 categorias de júri para avaliação dos nossos trabalhos, né? E esses avaliadores, então, que se disponibilizaram para realizar esse trabalho e dar essa grande contribuição para o nosso evento. E nós, gente, nós agradecemos sobretudo a você que está conosco agora, a você que desde o início está ajudando na divulgação, está acompanhando as nossas atividades, e que agora está recebendo o nosso encontro não mais dentro da universidade, mas está recebendo o nosso encontro na intimidade da sua casa, na intimidade do seu trabalho. Então, assim, esse é um momento muito especial. Nós não estamos unidos presencialmente, de forma física, mas estamos unidos em torno de ideais de um mundo melhor, estamos unidos em torno de um ideal do nosso Brasil ter protagonismo nas descobertas científicas, nós estamos unidos em torno de uma sociedade melhor. Então, que esse evento, né, seja uma amostra de tudo que nós podemos fazer por meio de um trabalho em colaboração. Eu desejo um ótimo evento a todos nós. E é com isso, então, deixa eu dar uma espiadinha aqui na minha aba do YouTube. Já estamos com 103 espectadores simultâneos. E, não, está sendo um sucesso, está sendo um sucesso. E aí o nosso evento, a nossa programação, então, como nós falamos, ela envolve a participação de pesquisadores americanos, canadenses e pesquisadores aqui do Brasil. Então, logo mais, às 19 horas, nós teremos a nossa palestra de abertura com o Dr. Enfu Rui, que é da Universidade de San Diego, da Califórnia. E às 20 horas, então, nós teremos a palestra com o professor Dr. Antonio De Mayo, também dessa mesma universidade, ambos professores, eles são colaboradores da professora Cristina Bonorino, e vão estar nos abrilhantando aqui com a sua palestra. Amanhã, quarta-feira, nós teremos uma palestra, então, com um colaborador meu, professor canadense, o Dr. Jean Sevigny, e que vai ser às 18h30 e às 19h30, então, nós teremos a palestra do professor, do Dr. Tiago Borges. Ele é um pesquisador brasileiro, ele foi formado pela professora Cristina Bonorino, mas ele está atuando no Massachusetts General Hospital, em Boston, que é um dos grandes centros de pesquisa dos Estados Unidos. Ele vai estar conosco amanhã. Na quinta-feira, nós teremos como tema Engenharia Tecidual. A primeira palestra ela vai ser ministrada às 18h30, então, pela professora doutora Veronique Moulin. A professora Veronique, ela é colaboradora da nossa docente, a professora Márcia Vinck. Recentemente, uma das suas alunas esteve lá para fazer um estágio, em torno de um ano, não me lembro exatamente se foi um ano ou perto de um ano, e a professora Veronique, então, ela trabalha com engenharia de tecidos. E, na sequência, nós teremos uma convidada que também foi escolhida a dedo por nós, que é a doutora Carolina Cagliari. A Carolina Cagliari, ela fez mestrado e doutorado pela USP, e hoje em dia, e após o seu doutorado, ela abriu uma empresa focada na questão de regeneração tecidual e em tecnologia de células-tronco. Ela é uma empresária de grande sucesso. No último ano, ela teve projeção nacional, e o nosso objetivo de chamar palestrantes com essa experiência é de despertar nos nossos alunos o espírito empreendedor, né? Nós temos muitos alunos, os nossos alunos são tão talentosos, então a gente vê que normalmente o aluno que termina o doutorado, muitas vezes fica focado na questão acadêmica, mas nós queremos apresentar outras oportunidades, nós queremos apresentar outros caminhos. Claro que cada um tem um talento, né, pessoal? Mas, enfim, para aqueles que têm esse espírito empreendedor e que têm o desejo de talvez ter a sua empresa, eu acho que essa palestra da doutora Carolina vai ser muito bacana. E no último dia, na sexta-feira, no último dia do nosso evento, nós vamos ter, então, a palestra, uma grande palestra do Dr. Jacob Glanville. O Dr. Jacob, ele é cientista, ele é imunologista, e recentemente ele fez uma série no Netflix sobre a pandemia. Então, assim, nós conseguimos uma agenda com ele, por meio de também um dos nossos alunos, aqui da universidade, um aluno do curso de farmácia, que acabou conhecendo ele, e que por meio do Giovanni é o nome, Giovanni Anesi, e que por meio dele, então, nós conseguimos o contato, ele vai dar essa palestra na sexta-feira. E após, nós teremos uma mesa redonda de discussão sobre divulgação científica, e essa mesa redonda, então, vai estar muito legal, porque ela vai ser composta por 3 grupos de divulgação científica. A doutora Melanie Dutra, que é do grupo Rede Covid, as professoras da nossa universidade, a professora Silvana, a professora Júlia e a professora... o palestrante chegou aqui. A palestrante... e a Marilu, e o Ciência Ponto Traduzida, né, que é das nossas alunas aqui do Biosciences, doutorandas Gabriele e Dominique. Então, nós teremos um momento muito proveitoso. So, I would like to... Christine is coming. I would like to give good afternoon to Dr. Enfu Rui. Thank you very much, Dr. Rui, to be with us in our in the Biosciences meeting. Christine is here also, and I would like invite to Christina to introduce you.
welcome, Dr. Rui. It's a pleasure having you here.
Hello. Hi.
Hello.
Hi, everyone. Can you hear me?
Yes, we can hear you.
Okay, great. Let me turn on my video.
Great.
You see me?
Not yet,
Well,
but it's coming. Not yet.
Yeah.
not yet. Okay. Finding webcams. Hmm. Hi. I think that it's working.
It's coming.
Hello.
Christine is here.
Hi guys.
Hi,
Hi
Kim.
guys.
Hi,
Hi.
Kim.
Hello.
Hello.
Hi. Hey Amfu, how are you?
I'm good. How are you?
Good. Nice to see you.
Yeah.
I got a message from Ricardo. Did you get it? About the
Yeah.
proteins?
Yeah, I got it.
That's
Yeah.
nice. I guess he was just busy writing grants, so it was a good thing that we waited, right?
Uh-huh. Yeah. Yeah.
I'm excited.
Starting his lab as well. So, you know how much work it is.
I know. That's okay. How are you? Are you excited? Everybody's excited to hear you talk.
Yeah, I'm excited. You guys have summer over there, right? Summer.
Yeah,
Cool.
starting the summer.
Yeah, it's a little too cold here.
Is it? California, come on, it's sunny.
No, there's no snow, but it still, feels a little too cold.
Really?
Yeah, my plants certainly don't like it. Yeah, no fruit. I have a couple of plant trees in the backyard.
In the backyard. What do you have? What do you have?
Actually, well, prominent. Is that how you pronounce it? And
Pomegranate?
Pomegranate. Yeah, pomegranate.
Nice.
And then peaches.
Nice.
Yeah, it's too cold. Too cold for them.
And Fu is a big fan of fruit. When he was here, when we were in Florianopolis, remember the meeting?
Yeah.
Years ago. He loved the fruit. He was like, this is too good. Yeah, this is a good thing.
Yeah.
It's a good thing about Brazil. We have a new president. Are you excited? I'm excited for you.
Oh yeah. Oh, so much relief.
So good.
It was difficult. Yeah, it was very close. At least it seems to be very close. There was some swinging state that really got the nerve of everybody.
Can you vote? Are you registered to vote?
No, I'm not a citizen.
Yes. I mean, I don't know if you want to do it, but
yeah, at some point maybe. Yeah. Like, I was worried about if Trump get reelected, whether I want to become a citizen.
No, he needed all the votes, all the possible votes. It was a close one. Yeah.
Mm-hmm.
So are you guys teaching? Are you still doing?
No, I'm not teaching right now, but I'm gonna teach in the winter. Actually, starting in a month.
Okay.
So yeah, will be another busy quarter.
Sounds good. Okay, so do we want to do
test? Yeah, test. How do I share my screen?
Elise.
I don't see the. Should I try to share my screen?
Yeah.
I make you the presenter. I make you the presenter of the session. So
the presenter. Okay.
In the bottom of your the screen will appear some a possibility to share your presentation.
Okay.
Can you find it?
I don't see the share button actually. I do see there's slides number 1, number 2, but I don't have control. I think it's your slides over there. But I do see a button on the left. Okay, let me see. What is this?
Don't you see an arrow on the bottom? An arrow?
An arrow?
I don't see an arrow, but I'm not the presenter.
I see a plus sign.
Yes, maybe plus, yes.
Upload the presentation?
Yes.
There's an upload. So that's different. I think that's
No, there is another possibility in the bottom. There is the first bottom from the right to the left. The first bottom.
Gurias, eu entrei no meio, nem sei se vocês já falaram com ele, eu nem apresentei vocês, mas eu entrei depois, eu não sei se vocês já tinham se apresentado. Apresento?
Não, agora você, agora falta a apresentação dele, Cris.
Tá, mas vocês, apresentar ele para vocês. Eu entrei no
mas
meio
a gente já está ao vivo aqui, não, a gente deixa...
A gente está ao vivo, Cris.
Ah, não sabia.
A gente está ao vivo.
Tá, e as perguntas vão vir, tu disse se vão passar para mim, tu vai mandar para o meu WhatsApp...
Não, aqui no chat.
Aqui no chat mesmo.
Aqui, vai passar aqui no chat as perguntas?
Sim.
Eu tenho medo que eu perca um monte de pergunta. Quem sabe passa para o meu...
Nós te ajudamos, Cris. Nós te ajudamos.
Ok.
Back. And I don't see the share screen.
Yeah, I'm doing now. And what about now?
What share your screen? Oh yes, I see that.
Falta minuto.
Share my screen, so it's interesting. And I can click the share my screen, but it's Chrome tab, maybe action window, your entire screen. So share audio.
Aqui na sala só tem pouca gente, mas no YouTube tu consegue ver quantas pessoas tem essas coisas? Não.
Tem. 110.
Não, só para a gente ter uma ideia.
Permission to share to access screen is not granted. Okay, so no, it didn't work. I can click share your screen, but then it's not my screen, which is weird.
O Antônio está perguntando se ele precisa de uma password para entrar no link, aquele que tu mandou. Não, né?
Não. Info, maybe you can send the presentation to me and then I can open for you.
I can try to upload it there. Upload.
Okay.
Do the upload then. Let's try the upload. I'm not familiar with this software.
Be better,
so.
I think.
Okay, let me try to upload it. Okay.
Yeah.
Okay.
I think that's probably the only. Browse. To be uploaded. I can. Upload.
Are you using Mac?
No, I'm PC.
Because
I'm uploading Urine files. I don't know whether the videos would work. I hope so.
Pois é.
I mean, I hope those animation will still work.
Estranho que ele não consegue share a apresentação. Consegue. Estou em Mac também. O Roberto, so we have the technical person is saying that he has a Mac also, so he'll probably be able to play your videos. It could be Safari. It could be because are you on Safari, or are you on what program are you using for this conference? Info. Are you on Safari?
PowerPoint.
No, but the
Oh,
internet.
I think it's maybe Google Chrome.
Is it Chrome? It should work.
Let's see.
Because,
yeah, Google Chrome.
Yeah, it should work.
Yeah.
For Google Chrome.
I mean, I'm happy to do. I mean, I guess it's too late to do a Zoom meeting. And I've been working with Zoom link for quite a bit. Oh.
Maybe it would be better to I can present for you and then you just talk. Maybe you can send to my email.
Yeah.
I think that
okay. I will. I'll do that.
But did you manage to upload it, Rui? Did you manage to do
it? It's uploading, then it says it's converting. It's converting to some other file. So I think, as you said, the sending you the email the file to you would be the best. So I'm doing that now. Hi, Lisandra. I just sent you the PowerPoint.
Okay.
Okay.
Did you get it?
Okay, so I guess what I'm gonna do is I'm gonna start presenting in full, and in the meantime, I'm asking also Roberto to try and contact Antonio because he's trying to log in and he doesn't know exactly how to do it. And I know Antonio sent his slides. So okay, so Roberto kind of figured out. So I'm gonna get started. Okay, so we're gonna get this info.
Go ahead.
Trust me, it's gonna work.
Yes.
Gonna work? Did you get my slides?
Yes, it's gonna. Okay.
So I will let you then. I'll say goodbye, and then I will be in the backstage now.
Okay.
Okay. Bye.
Okay. So Roberto is asking you and Fu to send it to his email. Roberto R. Can you do that?
What's his email? I sent it to Elisandra.
Okay. I think it's okay. So okay. So
I sent it to Elisandra. Elisandra, did you receive it?
Okay, we got this. This is gonna work. Yeah.
Okay. Okay. Excellent.
Okay. So I am so let me do this introduction because I'm dying to do this. This is one of my favorite researchers right here, guys. You're in for a really nice night tonight because we have two researchers that are going to talk in this session. I'm happy and I'm honored because they're both my friends. They're my colleagues. I've been working with them. Fu Hui is a professor at the Department of Biological Sciences, right? That's your department at UCSD. And Fu, I even wrote something down. I'm going to read it. He obtained his PhD in 2009 from University of Wisconsin-Madison. And he completed his postdoc with Ron Vale at UCSF. And Ron Vale, super nice guy. He worked a lot on cell biology of movement and cells, cytoskeleton. And Amfu, he is now with the Division of Biological Sciences at UCSD since 2016. And he was named a Searle Scholar and a Pew Scholar. So he's been through a lot of awards and collaborations. It's really nice work. And what he did with Ron Vale at UCSF was to contribute to a very, as I say, a very core problem of immunology. The signaling events that follow the engagement of the T cell receptor are very crucial to all of the immunological responses. And Amfu, when he was working at UCSF, he developed this system, which is an artificial system, where he reconstituted the events that happen, the biochemical events that happen when the TCR engages and signals. So it's the proximal signaling events of the TCR engagement. And it was a really nice paper, really cool to, everybody really talked about it. It's very cited. And then he went ahead and he used the same system in a different paper to show another part of the signaling events for T cells that are very important, which is the Pd1 signaling, which is a negative receptor that negatively controls T cell activation. So Enfu showed how the Pd1 signaling was affecting both the TCR and CD28. And then more recently, he's been using the same system that he's going to show us tonight to show cis and trans interactions, how they differ, the same receptors, if they interact in cis or in trans from cell to cell, how the signaling events are different and how that impacts the cell biology. And so we're very happy that you accepted this invitation tonight. We're very excited. And I hope you have fun tonight, and we're going to have questions for you afterwards. Welcome, Amfu.
Oh, thank you very much, Christina. I'm very honored to be speaking here to you guys. So I don't see the screen, the presentation screen for some reason.
Okay.
Just a moment, Amfu, that we are providing the sharing. Roberto is gonna solve this question. Just a moment, please.
Okay.
It's almost there.
Yeah.
Yes.
All right.
But maybe you can start telling the kids how you decided to build this artificial system. Why do you think that's important? What was the deal that made you work on that? Do you remember? It was a while ago.
Yeah. I mean, I think we wanted to. There were some questions that you can ask with animal models, with cellular systems, but the precise mechanisms sometimes can be challenging to ask in cells or in animal models. And so we wanted to use a bottom-up approach to try to assemble things from scratch, and to start with simple signaling molecules, signaling network, build a network so we know exactly what's going on in that system, and that give us control and offer the biophysical precision to figure out the detailed mechanisms of these signaling reactions. And so at the same time, we also, validate the findings that come up with this reconstitution system in cells or even in animal models. So we are working in between those cell-free system and the cellular system, and to come up with a more complete model to explain T-cell signaling. Yeah.
Yeah. So I guess what we're going to see tonight is how you did that and how we can use that to compare your very reductionist system to a cellular system, and then extrapolate to an animal, like a whole organism, and how that can work.
Exactly.
And here we go. Okay.
Oh,
took some
excellent.
time, but it's worth it. Okay.
Hi, everyone. Sorry for the delay. Good evening. And so let me just start by telling you what we have done in the past 4 years at UCSD. So we work on this so-called immune checkpoint molecules, as Christina just talked about a little bit. And those are basically brake molecules expressed on our immune cells. So next slide, please. Can I actually advance the slides? Okay, I think so. Okay, great. So so the big contact of our work is this T cells, which is a type of white blood cell that patrol our body, identify and destroy virus-infected cells and tumor cells. So what you're looking at here is a video from Gillian Griffith's lab, showing that a T cells is attacking this tumor cell, as shown in blue here. I'm not sure whether I can play this video. I guess it's not working, but that's okay. So basically, the T cells can interact directly with the tumor cells to form this so-called immunological synapse. So at this cell-cell junction, you're going to find a lot of ligand receptor interactions, and these interactions would trigger either positive or negative signal to influence the T cell activity. And T cell would be able to integrate those signals and make a decision about killing or ignoring the target cell. And all right, so here, all right, I guess the diagram is okay. So here, all right. I don't know whether this. I used to have an animation here, but it didn't work out, so it just played out everything. So don't be scared by this slide. So let me just walk you through these molecules. So there are 3 signaling pathways that dictate the T cell activity. Shown on top of the T cell, on the bottom is the target cell. The first one is the antigen-specific signal through the T cell receptor, or TCR, which bind to a peptide antigen presented by the MHC molecule on the target cell. And this binding interaction trigger a positive signal through the ZAP70 kinase to activate T cells. But oftentimes, TCR signal is not sufficient for T cell activation. Additional positive signal is required, and these are mediated by the co-stimulatory receptors. And the major co-stimulatory receptor is called CD28, which has its own ligand, CD80 and CD86, also expressed on the target cells. So this binding triggers another type of positive signal that activate different set of molecules to co-activate the T cells together with the TCR. And on the other hand, T cells also have these immune checkpoint or molecular brakes that restrict their activity. Just like your car having a brake, T cells also have brakes. And they not only have one brake, they have multiple type of molecular brakes. One of them is CTLA4, as shown here, which compete with CD28, the stimulatory receptor, for a common set of ligands. So normally CTLA4 can outcompete CD28 for this ligand because of the higher affinity. I will return to this or get to this point in a few slides. And so another very well studied immune checkpoint inhibitory receptor is Pd1, which bind to its own ligand PDL1 on the opposite site. And this binding interactions triggers inhibitory signal through the phosphatase SHP2 to turn off TCR and CD28 signal. There are several other immune checkpoints found on T cells, and co-expression of these inhibitory molecules is a defining feature of the dysfunctional, exhausted T cells in the tumor microenvironment. So basically, these inhibitory receptors serve to protect the tumor cells, to help these tumor cells to evade immune destruction. And because of that, these inhibitory receptors are key cancer immunotherapy target. Drug companies are making antibody that can block these inhibitory receptors for cancer immunotherapy. For example, anti-CTLA-4 and anti-PD1 has been approved by FDA for cancer treatment. And for PD1 antibodies, there's a different version, which is targeting the ligand PD-L1. So typically, they're thought to be equivalent because they both block PD-1, PD-L1 interactions. But hopefully, by the end of the talk today, I can convince you that these two versions of antibodies are somewhat different. There's also some synergy between this CTLA-4 antibody and PD-1, PD-L1 antibody. So the detailed mechanism is not clear. Okay, so these checkpoint blockade therapies is breakthrough in oncology, but it's not perfect. It only works in a small subset of tumors and only benefit a fraction of patients. So we need the better biomarkers to predict patient response. We also need to develop additional target or combination therapies to benefit the non-responding patient. But all of these requires a better understanding of these immune checkpoint molecules. It turned out that we know very little about their fundamental biology. So many labs, including us, are trying to bridge this mechanistic gap. So work in my lab can be divided into two axes. So we are interested in the intracellular mechanisms of immune checkpoint. We want to figure out the downstream effector molecules of the immune checkpoints, and how do the signaling pathway differ from each other? Why do we need so many breaks on T cells? And how do they crosstalk or synergize or compete with one another? And on the other hand, we are also interested in the mechanism that regulate checkpoint interactions. For example, what are the mechanisms that can regulate PD-1, PD-L1 interactions? So in today's talk, I like to focus on the second aspect of research. Okay, so these are the molecules that I just introduced to you. So basically, receptors on the T cells bind to their respective ligand on the target cells, and these binding either trigger positive signal or negative signal to reciprocally regulate T cell activity. So this is a sort of a textbook image, but in reality, it's much more complicated, as you can expect in real biology. For example, we know that the target cell can express PD-1. Oh, this is broken again. All right, so the target cell can express the receptor, and the T cell can express the ligand as well. And there's also ligand expressed on the T cells as well. So several ligands can be expressed by T cells. And so we wanted to ask the question whether the co-expressed ligand and receptor can interact with each other. insist on the same cell membrane. So all right. So structurally, this actually makes sense, because if you look at the co-structure of PD1 and PDL1, what you see is a L-shaped complex. They don't bind in a linear fashion. They bind to each other with an about 90-degree angle. So we think that this L-shaped complex would be compatible to this cis interaction as well. But how do we know whether the cis interaction indeed take place on cell membrane? We couldn't use a typical binding assay that occur in solution, because these assays doesn't have the membrane geometry, so they don't distinguish trans versus cis interaction. So Yun Long, a postdoc in my lab, came up. Oh, this is going to be difficult to explain. All right, so they. What should I do here? All right, so, well. So what we did, okay, let me try. Let me just try to pretend this diagram is good. Okay, we use a liposome to mimic the cell membrane. Okay, we came up with a strategy to attach purified proteins to the liposomes so that we can control their concentration. We can also label these proteins with fluorophores to measure their interaction in cis. So to make a long story short, is we found that these two proteins, PD1 and PDL1, indeed interact with each other in cis. Then the next question is, how does this cis interaction between PD1 and PDL1 modulate the trans interaction? Do they actually compete with one another? To address this question,
yeah, I can see the slides are not showing completely.
yeah, I don't know, where are they?
Yeah. Okay, but maybe we can just go ahead and try to explain, because, so let me just understand this. Are you sharing your slides, or who is sharing your slides exactly?
I don't know. I actually don't know who is controlling the slides.
Okay.
I can only.
Roberto is showing the slides.
Okay,
Yes.
but he's not. Now the slides are. Can you see how it's cut? They're not completely. Tá vendo que tá cortado o slide? Ele não tá inteiro. Só aparece um pedaço do slide. O Roberto tem que estar em tela cheia. Ele não está nos ouvindo, Roberto, nem nos lendo, porque a gente está chamando ele aqui.
Estou ouvindo sim. Estou olhando aqui já.
Tá, e tu não consegue botar a tela cheia, porque eu acho que o Enfu caiu, hein? Eu não tô vendo ele.
Não, ele voltou.
Ah, tá. Vamos tentar ajudar, né? Porque, ó, quando tu fez isso, apareceu o slide, quase. Ok. We're trying to figure this out, Enfu. Just take a little time. It's going to be okay. Okay, so it's getting there.
É isso?
Dá para voltar um pouquinho?
Okay, can I continue?
Yes, please.
Okay, so all right. I don't know how to advance the slide though. Can I just? Okay, now I can do it. All right, so we know that these two proteins bind to each other both in trans and in cis. So we wanted to figure out whether these two mode of interactions compete with one another. To do this, we use a cell bilayer hybrid systems in which we have a T cell interacting with a lipid bilayer, which mimic the target cells. Okay, the reason we use the lipid bilayer is that we can control the identity and the concentration of these ligands. So basically, this lipid bilayer is decorated with MHC peptide, which is the ligand for TCR, and PD-L1, the ligand for PD-1. So when we drop a T cells on top of the lipid bilayer, the ligand receptor can engage each other in trans. And when they bind to each other in trans, you can see that both PD-1 and TCR form microclusters. So these microclusters are basically a readout for the trans interaction. Okay, so we use this readout to evaluate how the cis interaction can interfere with the trans interaction. So at the bottom, what you see here in this condition is we reconstitute PD-1 to the lipid bilayer as well, to allow for cis interaction between PD-L1 and PD-1 on the same membrane. So in this case, when you look at the T cell PD-1, they don't form microcluster anymore, whereas the TCR still form microcluster. So this experiment demonstrated that the cis PD-L1 PD-1 interaction can block the trans PD-L1 PD-1 interaction. Okay, so this actually solved a puzzle that dated back to my postdoc career. At that time, I was trying to set up a cell culture system to study PD-1 signaling. I decided to use the OT1 killer T cells and EL4 lymphomal cells to co-culture them because this was a well-established model to study TCR signaling. If you load these lymphomal cells with peptide antigen, you can stimulate the TCR signal in the killer T cells and allow this activated killer T cell to kill these lymphomal cells. And you can use a commercial kit to measure the lysis of this tumor cells. So that serves as an indication of the T cell activity. We also know that this T cell express the inhibitory receptor PD-1 and the tumor cell express its ligand PD-L1. So in principle, this inhibitory pathway should be there to suppress this T cell activity. So if that is true, if we use antibody to block this interaction, we should detect the increase in the tumor lysis. But look what happened. When we add the PD-1 antibody, we don't see any difference in the killing, suggesting that something is missing in our understanding about this very simple co-culture system. So my postdoc Yun Long figured it out. He found that the tumor cell also expressed PD-1, and because it expresses this PD-1, this PD-1 can bind to this PD-L1 in cis to prevent the trans interaction to take place. All right, so basically what he did next was to use CRISPR technology to knock out the PD-1 from the tumor cells. Now he was able to detect the anti-PD-1 response. Okay, so unfortunately the slides doesn't play very well, so I wasn't able to show the interesting data here. So you can imagine that when you knock out the PD-1, you now see that it was able to make the system respond to PD-1 blockade antibody. Okay, so the take-home message from this data is that the cis interaction can block the trans interaction. So even though you have this expression of both receptor and ligand, they don't trigger inhibitory signaling because the ligand was blocked by the cis receptor. Okay. All right. So can you please go to the next slide? I'm not able to advance the slides from here. Hello?
Yes, just a moment, please, because excuse me, our technician is trying to make better your presentation. Just a minute, please.
Okay.
Okay, so what are we doing?
The slide is not moving.
Uh-oh. Okay.
I'd like to advance to the next slide.
Okay, so what he said was that he was gonna take your permission to present, but then he's gonna be able to play your videos. I'm so sorry, Info. I don't know what's going on.
No problem. I mean, it's hard to do remote presentation anyway. It's
give me a link. Let me see. He sent me an email.
I'm so sorry, Fu. Let's try to.
no problem. I understand.
Let's try to correct this situation. Para as pessoas que estão nos assistindo, eu peço um pouquinho de paciência, que a gente já vai contornar esse pequeno problema tecnológico aqui que a gente está tendo, tá? Só um momentinho.
Okay, I'm so sorry that happened. We really don't know, because this normally works. This is the link we use for all the presentations here, but we're going to figure this out. He says he's uploading it. He's going to do it. It's just taking a while because of all the videos, but it's going to work.
Okay, you guys are in the evening, so I have plenty of time. That's fine. I don't mind starting over.
I guess we can chat a little bit while they're doing this, because what we're trying to show, I don't know how many people are familiar with the PD1, PD-L1 axis, although it's pretty famous for all the immunologists. Like Enfu said, today it's a central problem in cancer, because all the immunotherapy for cancer is done through this axis. And what we're seeing here is like Info is telling us the story of how he figured out that the way people normally represent these interactions, right, Info, where the T cell expressing PD1 and the tumor, the lymphoma cell expressing PD-L1, is not the only way that it happens. That was pretty cool. Yeah, because he was able to show that it can happen in more than one way. And this is a complete game changer. I remember when I saw him present this the first time. Remember this, Info? And I went up to him and said, if you're right, because this is the deal. This interaction, PD1, PD-L1, is what we try to interrupt, to cut when we treat cancer, because this is a negative signal for the T cell. But it only works, the therapy in patients only works for about 25% of the people. And when I saw this, I told Info, if you're right, if this is true, what you're showing, you probably have a lot of explanations about resistance for therapy. So this is actually pretty cool. So I wonder, guys, Roberto, how are we doing with the file? Because it's so cool. This is such a cool presentation. I know it's...
I wish I could share from my side. That would make things 100 times easier.
Yeah, normally this works. I don't know why it's not working right now, but we're gonna figure this out. Okay, how are we doing, guys? And now it says that it's converting. So
I mean, I can also do a Zoom link. If I share everybody a Zoom link, I can present through Zoom. That would be much easier. All the students can join a Zoom as well. That might be, yeah. That's just, we can make it happen now.
okay, so he says if you want to share the screen, you can try. Can you see a link on the chat on your left?
On the chat.
Can you see a link?
Yeah, I see a link. Yeah.
So he says that maybe you can try and share through that link. I don't know.
I mean, the link is basically the same.
Yeah.
I'll see. Okay, this is a link as a Google Doc.
Yeah, it looks like it.
A Google Doc to share with using a Google Doc.
He says just click on this and present. Let's try that. Can you try that?
Click on a Google Doc link.
Yes, and then say share screen. Let's see if we can do that.
Okay. Okay. Yeah, I can present. Can everybody see my screen now?
Not yet, but then
Yes. Now
we're gonna. I think he has to shift the. How do we do this, Roberto? He already accessed the link. Roberto, ele já acessou o link. Como é que a gente faz isso agora? Ok, but we're not, a gente não tá vendo, né? Ele tá compartilhando. Did you do share screen?
you know, here I have a share button. I don't think it's sharing.
Button. You have to take off the PDF, I think. Quem é que tem que tirar esse PDF? É o Roberto ou o Info? Tá, então, Roberto, tira essa apresentação e deixa ele fazer o share. So if you do share screen, Info. Can you
share
do it?
screen in where? In a Google Doc?
Yes. Let's see if this is gonna work.
I'm not able to share screen through Google Doc. There's no share screen button. There's only a
he says
button. I can start presentation, but you don't see the presentation from your side.
It says there's a button on the side of your camera that says on the Google Doc link. Should be a button on the side of your camera. À esquerda ou à direita, Roberto? O botão. On your right, on the right of your camera, is there a button for share? A blue
button? Is it share button? Yeah, but then it asked me to share the link.
Okay.
Send the link to somebody.
É para ele mandar o link para alguém? É isso, Roberto? Ele está dizendo que quando ele clica no botão, diz para mandar o link. Okay, this is, you have to share on the miniature screen. Do you see a miniature screen? Mas, Roberto, é no botão ou na tela? Miniatura. Ou o botão é a tela? Ok, olha só, eu tenho um botão de uma telinha e eu fechei agora. Roberto. Do you see on your right a little screen on a tripod? Do you see that?
I see.
Something is happening.
Yeah.
Okay. Yep.
You see that? See my screen now?
Yes. Similar results in Treg.
Yes, that's okay.
Okay. So I think you can either go back to where we were.
So you see my screen now?
Yeah. Mas assim, volta a slide então, Roberto. So which slide do you want to go to? To the one before that? So he's
This is still messed up here. Okay, I mean, that's okay. I don't have to explain this.
Okay, so where do you want to go? Let's see, which slide?
Well, maybe here.
You want to start here? Perfect.
Yeah.
Okay.
So which slide are you seeing? Is this PD1 neutralized PDL1?
No, I see one with Preston Dennett. CTLA4 transendocytosis.
Oh, that's not okay. Well,
okay, I want to go back.
This is not how it works. I guess. Okay.
Tá, Roberto, não é esse slide, tá? Nós vamos achar o slide. Let's go see which slide. Do you remember the number? The slide number? Can you see the slides?
I can see the slides now.
Okay.
Maybe just go back to the.
Volta, Roberto.
Part. This is the second part.
Volta. Volta bastante. Pode voltar bastante. Yep. Vai voltando. Ok. Vai voltando. Vai voltando.
Go slide 20, maybe.
No slide número 20, provavelmente. Okay. This was the one that we were trying to look at. PDL1. More?
Okay.
Go back.
Yeah, that's fine. Okay, yeah, just the previous slides is fine.
Tá, vai mais para frente. There?
Previous.
Antes desse. Mais vai antes. Antes. Aí.
Okay, yeah, this one. So on the right, what you see is when we knock out the PD1 from the tumor cells, we can recover this PD1 signaling. So when we block the PD1 pathway, you can see the antibody effect. Okay, so the next slides. Can you please go to the next slides? I have no control here.
Yeah. Passa o próximo, Roberto. There you go.
All right, so let me just summarize part one of the talk. We found that the PDL1 and PD1 can interact both in trans and in cis. And this cis interaction can serve to sequester the ligand from binding to the receptor in trans. So next, please.
Próximo.
And the implication about this work is, what kind of whether PDL1 is a good biomarker for PD1 therapy. At the moment, PDL1 is the most commonly used biomarker for PD-1 targeted therapy. What I mean is that doctors usually look at the PD-L1 expression in the patient, in the tumor samples. If the patient expresses PD-L1, that often indicate there's pre-existing PD-1 signaling going on. So this patient would be eligible for PD-1 therapy. But what we found is one proposed that even PD-L1 is expressed, it may not be functional because the cis interaction might serve to block the ligand, prevent the ligand from activating the receptor on T cells. Okay, next, please.
Próximo slide, pass.
All right, so then we want to tell you another study that focused on the PD-L1 CD80 interaction. So this was initially found to interact with each other. Okay, these two proteins interact with each other in solution. If you purify the extracellular domains of PD-L1 and CD80, they bind to each other. This was originally reported by Arlene Sharp, one of the pioneers of PD-1 signaling, PD-1 function. Initially, it was thought to bind to each other in trans across the immunological synapse because both PD-L1 and CD80 can be expressed by T cells and the target cell. So again, we wanted to fit. Next slide, please.
Próximo slide. Roberto, tu precisa ficar ouvindo quando ele fala next.
And I want to say this is a wonderful collaboration with between us and Christina's lab and Li Fan Lu's lab at UC and Jack Buey's lab at UCSD. It's a teamwork. We want to figure out the biochemical consequence and functional consequence of the PD-L1 CD80 interactions. Next slide, please.
Próximo slide.
All right, so in this case, because slides is messed up again, so we developed the reconstitution system to study, to test both trans interaction and cis interaction. So we want to figure out whether these two proteins bind in trans or in cis or both. And so we developed these cell-free systems to test either trans interaction or cis interaction. In this case, is a trans interaction assay. We prepare our liposomes, as shown in green here, and the supported lipid bilayer. We attach PD-L1 and PD-1 to the liposome and the lipid bilayer respectively. So we can incubate the liposome with a lipid bilayer and wash away the unbound liposomes. We can measure using TURF microscopy how many liposomes remain bound to the lipid bilayer. So this gives us a handle to measure the trans interaction. Okay, you can see that these green dots in the bottom, that's what you see under microscope. So each of these green dots is a liposome associated with the lipid bilayer. So this is expected because we know PD-L1 and PD-1 can bind in trans, right? But when we replace PD-1 with CD80, we don't see the liposome anymore, very few liposomes left. So this basically suggests that PD-L1 and CD80 do not interact in trans as originally proposed. Okay. Next slide, please. But we do have evidence from multiple assays that these two proteins interact in cis. So here I'm just showing you one assay we developed, and this is a cellular reconstitution assay. This is the HAC293 cells, the non-immune cells. So we transfect PD-L1 and CD80 to the HAC cells, and we label PD-L1 with a donor fluorophore and CD80 with an acceptor fluorophore. And using confocal microscopy, we were able to detect a strong energy transfer threat between the PD-L1 and CD80, suggesting that they bind to each other in cis on the same cell membrane. In fact, when we replace PD-L1 with PD-L2, a second ligand of PD-1, we don't see the threat anymore, very low threat. So this is the negative control. On the right is the negative control. So this data suggests that PD-L1 and CD80 bind to each other in cis. Next. Can you play? Okay, good. We also found that this cis interaction can be disrupted by PD-L1 antibody. This atezolizumab is an FDA-approved PD-L1 antibody. We found that in the presence of atezol, the threat signal was greatly reduced, suggesting that the PD-L1 CD80 cis interaction can be disrupted by PD-L1 inhibitor, PD-L1 antibody. Next, please. So these data collectively demonstrate that PD-L1 and CD80 bind only in cis but not in trans. Next, please. So we think that these two ligands actually form heterodimers on cells. And next, please. And so we also found that this heterodimerization of so we also found that there's two distinct effects of PD-L1 antibody. First, these antibodies can disrupt the PD-L1 PD-1 interaction. Okay, the trans interaction between PD-L1 and PD-1 can be disrupted by this antibody. Can you click the next? See whether it plays out. Okay, so this antibody can disrupt this trans interaction between PD-1 and PD-L1. Next. We also found that this antibody has a side effect, and this antibody can disrupt the cis interaction between PD-L1 and CD80. Yeah, can you click next? Yeah, so this antibody can disrupt the cis interaction as well. Can you click next, please? So these are inconsistent. These are consistent with recent work from Gordon Freeman's lab. They use completely orthogonal assays to demonstrate that PD-L1 and CD80, which is also called B7, only interact in cis on the same cell membrane, but not in trans as originally proposed. Next, please. So our next goal was trying to figure out the functional consequence of this cis interaction between PD-L1 and CD80. We wanted to ask whether this heterodimer can interact with PD-1, CTLA4, or CD28. And this is not a trivial question because there's no reported crystal structure for PD-L1 CD80 complex. There's not even structure for CD28 CD80 complex. So it's difficult to predict how this cis interaction would affect the trans interaction. And so we decided to use cell line assays, binding assays, to tease apart the three pathways, to look at one pathway at a time. Okay, next. So again, so there's no structure information, so we have to use binding assays to figure out one pathway at a time. Okay, next, please. So this is looking for PD-1 binding. So we generated two types of Raji B cells. That on the left you see a Raji B cell co-expressing PD-L1 and threefold excess of CD80. So these two can bind to each other in cis. On the right is a Raji B cell expressing only PD-L1, no CD80. And we challenged both cells with equal amount of PD-1 extracellular domain. And then we use flow cytometry to measure the binding of PD-1 to these two types of cells. As you can see in the flow cytometry diagram, when you have CD80 co-expressed, the binding of PD-1 was greatly diminished as compared to the no CD80 condition, suggesting that CD80 can inhibit PD-L1, PD-1 interaction. Next, please. We also confirm this finding using this cell culture system. When we incubate Jurkat cell expressing PD-1 GFP with a Raji B cell expressing PD-L1 and cherry, you can see that both PD-1 and PD-L1 goes to the synapse because of their trans interaction. And when we co-express CD80 with PD-L1, you can see that the enrichment of both PD-1 and CD8 and PD-L1 were decreased. Okay, so this demonstrates that the cis CD80 can indeed inhibit trans PD-L1, PD-1 interaction. Next, please. We also have evidence that the cis CD80 can prevent PD-1 signaling, prevent PD-1 phosphorylation. This was based on, again, a co-culture system, but using co-IP and Western blot as a readout. So Xiaozheng, a postdoc in my lab, used 3 types of Raji B cell to stimulate PD-1 expressing T cells. These cells express similar levels of PD-L1 by increasing levels of CD80. As no CD80, you see strong phosphorylation of PD-1, and low CD80, you see similar phosphorylation of PD-1. But when you have high amount of CD80, the PD-1 phosphorylation disappears. And same happens with the SHP2 recruitment. So this suggests that the cis CD80 can block the PD-1, PD-L1 interaction. Next, please. And this is consistent with Taku Okazaki's group, which provide evidence that CD80 can physically block the PD-1 binding interface on PD-L1. So this is a crystal structure of PD-1, PD-L1 complex, and the Okazaki group used a random mutagenesis to identify this tyrosine residue on PD-L1 is required for CD80 binding. Even though there's no structure for PD-L1 CD80 complex, they were able to show that this tyrosine is required for CD80 binding. As you can see here, the tyrosine is sitting, is also part of the PD-1 binding interface. So this means that CD80 is physically blocking the PD-1 binding. Next, please. So in summary, this part of our work suggests that the cis CD80 physically block the PD-1, PD-L1 interaction. So this heterodimer can no longer bind to PD-1 anymore. Next, please. So we next want to focus on this CD28 axis. We wanted to ask whether this PD-L1 CD80 heterodimer can bind to CD28. Okay. Next, please. So again, we use this binding assay. In this case, on the left, you have a double positive cells expressing CD80, both CD80 and PD-L1, but PD-L1 was 6-fold excess of CD80. So there was cis interaction in this case. On the right, we used atezolizumab to block the cis interaction. and we challenged both cells with CD28 extracellular domain. As you can see here, the binding was equivalent. So this suggests that the cis interaction does not affect CD80, CD28 interaction. Next, please. We were able to confirm this finding using cell co-culture assays. When we incubated a Jurkat cell expressing CD28 with a Raji cell expressing CD80, both CD28 and CD80 enriched to the synapse. And when we co-express PDL1, six-fold access to CD80 on the Raji cells, we still see the enrichment of CD28 and CD80. What is interesting is that the PDL1 was also enriched to the synapse. If you look at the bottom right panel, the PDL1 was also enriched, right? So I also want to point out that this Jurkat cells, or the T cell, didn't express PD1. So the enrichment of PDL1 was merely due to its interaction with CD80 and CD28. So we think this three proteins form a tripartite complex at the immunological synapse. Okay, next, please. And we know that this complex of CD80 and PDL1 can activate CD28 signaling just as well as free CD80. This is just showing that, you know, when you have no PDL1 or high level PDL1, CD28 was equally phosphorylated, was equally triggered by CD80. Can you see my cursor or a mouse? Probably
we can't
not.
see your, we can't see your cursor, but
you can or you cannot.
No, we cannot. But
okay.
What do you want to point to? What do you want to point to? Okay,
no problem. I'll keep that in mind. So I'll try to explain. Okay, next slide, please. These data suggest that the cis PDL1 has no effect on CD28 CD80 interaction. So so we think that their CD28 has two forms of ligand. You have the isolated CD80. You also have the CD80 PDL1 heterodimer as the ligand. Okay, next, please. So next, we want to look at the third axis, the CTLA4, whether PDL1 can interfere with CD80 CTLA4 interaction. Next, please. So again, we use this binding assay. We created two conditions. On the left is a double positive cells with cis interaction. On the right, the cis interaction was blocked by artiso. And we look at the CTLA4 binding. You can see that in the presence of cis interaction, the binding of CTLA4 was reduced, as shown in the yellow histogram in the middle. Okay, so the PDL1 seems to inhibit CD80 CTLA4 interaction. Next, please. So in summary, we found that this PDL1, the cis PDL1, selectively reduced CD80 CTLA4 interaction without touching the CD80 CD28 interaction, which is very surprising to us. Next, please. Because structurally, it doesn't seem to make sense. Previous biochemical studies indicate that CD28 and CTLA4 bind to the same face of CD80, and PDL1 bind to the other face, the opposite side of CD80. So it didn't make sense that PDL1 can selectively inhibit CTLA4 binding to CD80, but not affecting CD28 binding to CD80. Next, please. So to make a long story short, I just tell you the conclusion we found. So Yunlong, postdoc in my lab, he was able to dig deep in the literature and found that CD80 actually form homodimers. And this is a non-covalent interaction. But you can imagine on the antigen-presenting cells, CD80 exists in dimer and monomer in an equilibrium. Okay, there's a monomeric CD80. There's also dimeric CD80. Next, please. But this is important for distinguishing CTLA4 and CD28 binding. CTLA4 bind to CD80 homodimers, and it actually cross-links CD80 homodimers to form this lattice-like complex with very high avidity. They're holding their hand one by one, so to create a highly stable complex. Whereas CD28, as shown on the right, bind to CD80 in a monovalent fashion. So it is because this avidity, high avidity of CTLA4, that allow it to outcompete CD28 for the ligand CD80. Okay. So using a series of biochemical assays, Yunlong found the PD-L1 can inhibit CD80 homodimerization. Can you click the next, please? Next, please. Yeah, so when PD-L1 is round, PD-L1 would form heterodimers with CD80. Can you click next, please? Oh, no, okay. It doesn't play out. All right, so basically when PD-L1 form interact with CD80 in cis, it prevents the homodimerization of CD80, so that PD-L1, CTLA4 can only interact with CD80 through this low avidity dead end complex, as shown on the right. So the avidity of this CTLA4 interaction was reduced. Next, please. Whereas for CD28, it binds to CD80 in a monovalent fashion, so it didn't care about the presence of PD-L1. The PD-L1 has no effect on CD28 interaction. Okay, next, please. So we next want to figure out whether PD-L1 can modulate CTLA4 function, because we use in the binding assay, we found that PD-L1 can inhibit CTLA4 interaction with its ligand. What about at a functional level? Okay, next, please. So here I want to introduce you a very interesting type of T cells called regulatory T cells, because that's the major site of CTLA4 action. So normally when we talk about T cells, as I mentioned in the introduction slides, we talk about this conventional T cells, T conventional, okay? These are the cells that act as soldiers to destroy those target cells, okay? But this process doesn't happen spontaneously. Next, please, can you click the arrow? Okay, good. This conventional T cell require the priming, okay? Priming mediated by professional antigen-presenting cells, or APCs. Those are the dendritic cells or macrophages. They can do this job, priming and the conventional T cells. And next, please. And the regulatory T cells serve to inhibit this priming step. Next, please. So inhibit this priming step, okay, to reduce the conventional T cell activity. You can see that this, okay, next, please. And at a molecular level, this priming step required CD80, CD28 interaction. So this co-stimulation signal is essential for this priming step. So next, a CTLA-4 on the regulatory T cells are able to steal CD80 or deplete CD80 from the APCs. Next, please. So, okay, it didn't play out again. So that's okay. So the CTLA-4 is a very interesting molecule. It's highly endocytic. Okay, it largely exists in intracellular vesicles. Okay, next, please. So this is a confocal image of a CTLA-4 expressing T cells obtained by my student Preston Dennett. And you can see that most of the CTLA-4 exists in the vesicles, okay? And they constantly recycle between the intracellular vesicles and the plasma membrane. So at the plasma membrane, it can bind to CD80, okay? And then when CTLA-4 is endocytosed, it can pull CD80 away from the APC, which is an amazing process. And the cell biology is not clear, but we know that it happens. So it can deplete CD80 from the APC through this so-called transendocytosis process. Okay, next, please. And Preston was able to obtain visual evidence for this transendocytosis activity. So he incubated T cell expressing CTLA-4 with the Raji B cells expressing CD80 GFP. Okay, so you can see that while CTLA-4 exists in these intracellular vesicles, the CD80 can actually move from the APC, the Raji side, to the T cell side. If you focus on the image in the middle, the CD80 channel, you can see that CD80 goes to the T cell as well. It starts off on the APC side, it goes to the T cell, and the CD80 coexists with CTLA-4 in those intracellular vesicles on the right, the merged image. So this indicates that this is due to the CTLA-4 transendocytosis ability. Okay, we wanted to figure out whether the PD-L1 can interfere with this process, because we know that the PD-L1 can inhibit the CTLA-4 CD80 interaction. Next, please. So we set up a flow cytometry assay to quantitate this process, the CTLA-4 transendocytosis. And on the top panel, we have a CTLA-4 expressing T cells and form co-culture with CD80 expressing Raji cells. And we're using flow cytometry to monitor the CD80 level. Okay, at 0 time point, 0 hour, you see that the CD80 level is very high on the Raji side. But after 12 hours, most of the CD80 was depleted. Okay, the x-axis is the CD80 level. Okay, so this is because of CTLA-4 activity. It depletes CD80 from the Raji APC. And on the lower panel, what you see is when we co-express PD-L1 with CD80 on the Raji side, the CD80 depletion was inhibited. So CD80 level can be much better preserved after 12 hours. And we know that this is because of PD-L1 CD80 cis interaction, because, click next, please. When we add the PD-L1 antibody atezol to disrupt the cis interaction, now the CD80 depletion was restored. So we think this illustrates a side effect of this atezolizumab. It can deplete CD80 from the APC. And next, please. And as expected, this side effect can be blocked by CTLA-4 inhibitor, ipilimumab. So when we block CTLA-4 function, the CD80 level can be restored. Next, please. So in collaboration with Li Fan Lu's lab at UCSD, we were able to confirm this finding in Treg dendritic cell co-culture systems. So in the absence of regulatory T cells, the dendritic cells express high levels of CD80. And then next, please. When we have a Treg around, there's only a small decrease in CD80 level, despite that Treg express high level of CTLA-4. So this is because the DC also express PD-L1, that the PD-L1 bind to CD80 and protect CD80 from the Treg activity. We know that because when we block the PD-L1 using antibody, next please, you can see that the CD80 level was further reduced, okay? And then this side effect can be blocked by the CTLA-4 antibody. Next, please. You can see that co-treatment of CTLA-4 antibody recover the CD80 level, as shown in the yellow histogram. Next, please. And in collaboration with Christina's lab and Jack Buey's lab at UCSD, we also confirmed this finding in mouse tumor models that when we treat PD-L1 antibody, the CD80 was depleted from both macrophages and dendritic cells. Okay, but this depletion was not associated with anti-PD-1 treatment. So we think this side effect is selective to PD-L1 inhibitors. Okay, next, please. So I want to use this diagram to summarize part 2 of the talk today. So we think that on the antigen-presenting cells, APCs, there are 2 forms of ligand. You have the PD-L1, isolated PD-L1 as shown on the left, the gray molecule. You also have this CD80 PD-L1 heterodimer, okay? And these 2 forms of ligand has distinct activities. Only the isolated PD-L1 can bind and trigger this PD-1 inhibitory signaling. And the heterodimer does not bind to PD-1 anymore because the binding site of PD-1 is physically blocked by CD80 in cis. However, this heterodimer can bind and activate the CD28 co-stimulatory receptor. But this heterodimer only weakly binds to CTLA-4 because it doesn't have the avidity required for high affinity interaction with CTLA-4. But when you treat, next please, when you treat the system with PD-L1 antibody, there are 2 things that's going to happen. One is that this antibody would disrupt the PD-L1 PD-1 interaction to terminate the inhibitory axis. The second is that this antibody would also disrupt the heterodimers of CD80 and PD-L1. This would free up CD80 to form homodimers, as shown on the right, bottom right. Those CD80 homodimers would then interact with CTLA-4 and cause the depletion of this CD80 from the antigen-presenting cells. So there's 2 opposing effects of this PD-L1 antibody. Next, please. Okay, so in summary, we have found that there's 2 types of CD28 ligand. There's isolated CD80. There's also CD80 PD-L1 heterodimers, which is new finding. And then we also found that there's 2 functions of CD80. typically CD80 is known as the CD28 co-stimulatory ligand. And we found that CD80 can block PD-L1 function in cysts. Okay. We also found that there's two functions of PD-L1. It is well known to be a PD-1 ligand, but we found that PD-L1 can actually restrict CTLA-4 activity by protecting CD80 from CTLA-4 mediate depletion. And along the same line, the PD-L1 blockade antibodies would stimulate the CTLA-4 activity, and this side effect can be prevented by co-blockade of CTLA-4. We think this has implications of the synergy of CTLA-4 inhibitor and PD-L1 inhibitors observed in vivo. Okay, next please. All right, so I just want to thank the people who did this work, my very talented postdoc, Yunlong Zhao, Xiaozheng, and students, Preston, and our amazing collaborators who come from a very different background and provide complementary expertise that raise our work to the next level. So also thanks my funding sources and for trusting my idea. And thank you very much for you to stay with me. And so I'd like to take any questions. Hopefully this is not too disruptive. All right.
Okay, thank you, Anfu. Thank you so much for your resilience, because you didn't give up on us. And I'm sure that everybody's saying here, amazing talk. I'm just getting the comments here.
Yeah.
Just getting the comments. Thank you so much for doing this. And this is a very complicated problem. And every time I hear about it, I find something new. Like I get a new aspect that I hadn't thought of. Even though I work with checkpoint blockade, but I work more on the clinical side or preclinical side. So this is so intricate, the way the molecules can be working. So I have a couple of questions while we wait for some others that I was thinking about. But well, one is the implication that you have these two ligands, as you say, for CD28, which one is CD80 and the other one is the dimer, the heterodimer. Well, there's also a third ligand that we didn't talk about today, which is CD86, right? And we know, because we've worked on this, that CD86 is different. CD86 doesn't do the same things that CD80 does. So we're talking about a receptor, CD28, that's crucial for stimulating T cells. And we're talking about 3 different kinds of ligands, depending on the physiological state of the cell and depending on timing. I mean, do you have any idea how this plays in vivo in different cells? Can you even imagine a model where things are going on sequentially and this is happening through a process where a T cell is being activated? Or do you think this are going to be situations that the T cell is going to find in different tissues? Or how do you imagine that? I mean, this very
that's an excellent question. You talk about CD86. We didn't work too much on CD86, but it doesn't bind to PD-L1. So I think there are 2 major differences between CD80 and CD86. First, CD86 doesn't bind PD-L1 like CD80. Second, it's a weaker ligand for CD28. So I think that if CD80 and CD86 are equivalently expressed, CD80 would be the dominant ligand for CD28. And because CD86 is not protected by PD-L1, so it's probably more susceptible to Treg mediate depletion. inhibition. And David Sansom and others have shown that CD86 can be transendocytosed by CTLA4. And I think that that also depend on the relative expression kinetics. Which ligand comes first. When DC maturate, expression levels increase for both CD80 and CD86, I believe. And the same thing happens for PD-L1. In the inflammation condition, the PD-L1 is often higher than CD80. So, and several labs has reported that. So we think that PD-L1 protection effect for CD80 would be pretty significant in the context of inflammation. So in other words, I think that it's going to be hard to find isolated CD80 in that context because of the excessive amount of PD-L1. But the in vivo, what condition and what condition, I think it's going to be context dependent. Which one is more dominant than the other? Yeah.
Yeah, when you think about all the infiltrating cells in the tumor and that they all can express differentially I have other questions here for you. This one is from Thiago. He wants to say amazing talk. He's saying, what leads the APCs to express CD80 mono versus the homodimer? The homodimer or just the monomer, or CD80 heterodimerized with PD-L1. Is the signal through a TLR? If I use different TLR ligands, would I see different ratios of CD80 mono versus homodimer?
A great question. Unfortunately, I don't have the answer. I think the TLR axis is an interesting angle to study. So to answer your first question, what leads to express homodimer or mono CD80? I think the cells doesn't know. They don't have a preference which form of ligand they would express. They just express CD80 display on the cell membrane.
Probably dynamic.
Intrinsic affinity with each other. So once you have CD80, you're going to have homodimers. And the more CD80 you have, the more homodimers you have, right?
Makes sense.
It's driven by mass action, right? This is equilibrium. I think it would be valuable to study the upstream signal that induced expression of CD80 and PD-L1. I think that would be very valuable to the field. Yeah.
There's probably a kinetic too. There's another question. In the clinic, we use CTLA-4 Ig for autoimmunity and transplantation. So now we're doing CTLA-4 Ig with very exciting results. If the CD80 monomer or the CD80 PD-L1 heterodimer activates CD8 and only the homodimer binds CTLA-4, how could this drug be working?
Okay, let's see. First, I want to say, good, very deep question. The CTLA-4 still bind to the heterodimer. I think I didn't make it very clear throughout the talk, partly because of the slide problem, but we have evidence that it still binds. It just doesn't bind in a high avidity fashion. It still binds because the binding site is preserved. Okay, it just can't form this cross-linked lattice. It reduced the avidity of the interaction, but it still binds. So
it should bind to the dimer. It should bind to the dimer.
It binds to the homodimer better than the heterodimer, but it still binds to heterodimer. So the way that CD80 inhibit PD1 binding is very different from the way that PD-L1 inhibit CD80 CTLA-4 binding. The first one, the CD80 is physically blocking the PD1 binding site. Okay, it's blocking effect. And the CTO4, the PDL1 effect on CTO4 is not a blocking effect. It just reduced the strength, reduced the ability of the interaction.
So the drug would still be binding. The drug would still be binding.
Yeah.
So
I think CTL4-IG would work by occupying CD80 and CD86. It could be due to CD86, first of all. It could be due to occupying any form of CD80, right? Because the epitope of CD80, the CTL4 binding epitope on CD80 still preserved when PDL1 binds to CD80. So CTL4-IG will still bind to that heterodimer or homodimer or monomeric CD80 or CD86.
Could still be blocking CD86.
Yeah, it just block the CD28 pathway.
It's probably something else goes on, but I mean, there's still room for investigation, but it's probably that's what's happening. And my last question, because I really, we're gonna move to the next one, but so we have a problem now if this works, if atezolizumab can have these effects and ipilimumab can solve them. I think we talked about this. They're made by different companies. So I think it's Roche, right? Atezo is Roche, and ipilimumab is Bristol Myers. So they would have to come up with a clinical trial together. Have you ever talked to them about that? Have they talked to you about that?
I'm not. No, I haven't talked about that with them. That's an interesting idea. There are existing clinical trials with PDL1 inhibitor and CTLA4 inhibitor. There are ongoing ones.
I think there is. But it's interesting. I mean, anyways, I would like to see that.
In general, it's fair to say that the PD1 inhibitor is better than PDL1 inhibitor in those clinical trials.
That's true.
And also in mouse tumor models, Jim Allison has this PNS paper in, I think, 2013, comparing PD1 inhibitor and PDL1 inhibitor. And the PD1 inhibitor was better than PDL1 inhibitor. But one has to also consider the binding affinity of these antibodies. Maybe the PD1 inhibitor has a higher affinity than the PDL1 inhibitor. But there could be many mechanisms behind those differences in therapeutic.
The binding site, for one, the binding site is a given. We don't know enough. But... Okay. Well, thank you so much. Thank you so much for sticking with us. Thank you so much for this amazing talk.
Fun experience.
I'm getting a lot of compliments here on the chat. So, I mean, thank you so much. This is great for our students. This is great for our professors. I'm sure that everybody enjoyed it. And I can't thank you enough. Thank you so much.
Oh, thank you. Thank you, everybody, for staying with me.
Okay. So I can see we already have our next presenter online.
Hi.
Yes. Okay. Thank you very much, Ifu. Thank you.
Everybody.
We have more than 100 students and researchers seeing you in our YouTube
I look forward to that.
People love your presentation and your research. Thank you so much. And sorry for this technological problem that you had tonight, but in the end, everything was all right. And
it's fun experience for me.
Thank you so much. And thank you, Antonio, for coming with us now.
Okay, talk to you later. Bye.
Okay, so are we gonna go? Are we a go for Antonio's presentation?
How do I share the slides?
Can we see the slides, and then when we have the first one, I'm gonna introduce you. Because
we already made the upload of your presentation, Antonio, just to avoid problems. And then our technician is going to help us to share the presentation to the participants. So thank you so much, Antonio.
No
okay,
problem.
so can I go ahead then and introduce Antonio? Is that fine? Is that okay? Are we ready? I can see you're in the lab probably. Are you?
Yeah, I'm in my
Yeah, I
office.
can see that.
I'm very
So-
professional.
You are.
And I'm not wearing pajamas, you know?
You're not wearing pajamas because you have the Self Stress Society shirt on. So guys, I'm very happy to introduce to you tonight Dr. Antonio De Mayo. Dr. Antonio De Mayo is my friend, my gracious host. When I go to UCSD, I go to his lab. We have endless conversations in differential things. We also have parties. It's very exciting. Antonio is actually from Venezuela, as he likes to say. Has very, I'm sure, very nice stories that he can tell us about it. But he actually went to Israel. It's a very interesting story because he went to Israel to the Weizmann Institute, and that's where he got his PhD. And then he joined UCSD, San Diego, the University of California, San Diego in 2006. But before that, he was the research director for the Division of Pediatric Surgery and associate professor at the Johns Hopkins University in Baltimore, where he forever became bound to their football team, the Ravens. And while at Hopkins, Dr. De Mayo led the committee for the recruitment of underrepresented minorities to graduate programs and the post-baccalaureate research education program, which is called PrEP. At UC San Diego, Antonio is very engaged in increasing student diversity in graduate and medical school. Antonio is never tired, always willing to increase diversity. He's been helping us to do a collaboration between UFSPA and UCSD. And Antonio has has a diverse interest in research in his lab. He is investigating the molecular and genetic basis of the response to injury. So trauma is a big interest for Antonio. The other interests are stress proteins, cell stress. So that's how we met in cell stress meetings. And he lately has been studying a lot of extracellular vesicles and the biology of these vesicles in the intercellular communication. And is going to show us, I'm sure today, some very interesting insights in the lipid composition and other aspects of extracellular. Thank you so much for being with us tonight, Antonio. We're very happy to have you.
Now that we're talking about the Ravens, we need to finish by 5 because they're playing tonight.
Oh my God.
So thank you so much for inviting me to this conference. Obviously, the sad part is that I would rather be in Brazil eating, drinking caipirinhas with all of you.
Yes.
But, this is what happened in this time. So let me start with this slide because I want you to pay attention. Let's see if it works. Yes, please pay attention to what I'm going to say because I'm not going to repeat it. So I like to start this talk with this phrase of Paracelsus that say that the body possesses the high art of recalling and also restoring. And this is exactly what the organisms and the stress responses that at the same time that can cause a harm can also have the machinery to restore the harm. Now, organisms and cells are constantly exposed to changes in the environment that could be a stress, which may compromise homeostasis and cellular integrity. So we all know that there are many different types of stresses. You have environmental stresses like temperature, radiation. You also have chemical stresses like heavy metals, like high amount of zinc or stanium or arsenate. There is also a stress related to infections like viruses, parasites, bacteria. And finally, there is also a mental stress. So a hallmark of the cellular response to stress is the expression of Hitchcock proteins or stress proteins. And these proteins participate in the repair and recovery from different kinds of insults. And after their expression is achieved, these cells became tolerant to a subsequent stress, what is called stress tolerance. Now, also, we know that Hitchcock proteins are also present during normal conditions, and they participate in different cellular processes, particularly cellular protein folding. The role of Hitchcock protein is very important in folding nascent polypeptides, and they are, in general, considered molecular chaperones. And I actually writing a story about what this terminology was coming about. Now, we know that there is no one Hitchcock protein. There are many Hitchcock proteins. There are different families. They are classified according to the molecular weight, HSP90, HSP100, HSP70. The HSP70 is the most recognized for everybody. And let me emphasize something about HSP70. There are 4 in mammalians. There are 4 HSP70s, and they are. One of them is exclusively inducible, so it's not present in normal cells, and the other 3 are constitutive, but present in different subcellular compartments. One in the cytosol, one in the ER, and another one in the mitochondria. Okay, so although all Hitchcock proteins are expressed within cellular compartments, they are also being found outside cells, and that's a new trick that we found with the HSP70, so the Hitchcock proteins. Now, the discovery that Hitchcock proteins were outside cells were for this guy, Larry Hightower, and his student Peter Didon, that in 1989 found that HSP70 was released from cells by a mechanism that cannot be blocked by conventional inhibitors of the classical secretory pathway, and it was not due to cell death. And in addition, they found that the protein was associated with fatty acids. That is another important thing. This discovery was forgotten for many years, and that's what I call Larry the Christopher Columbus of the extracellular Hitchcock proteins, because he discovered the proteins, but he didn't know where he thought he was still in India. So extracellular Hitchcock proteins are involved in many diseases. This is a very partial slide, but you can see that they are present in the extracellular space after cancer, acute injury, diabetes, infection, and many other cases, trauma. So they can be think as potential biomarkers, and that have been one of the interests in the extracellular proteins. But they also have a function. Okay, and let's start with this. There are 2 sources of extracellular Hitchcock proteins. There is an obvious passive mechanism in what the proteins that are contain HSP70 became necrotic and release all these cellular components outside the cells. So now you can find HSP70 outside the cells, but it's because it's due to cell death. However, there is an active mechanism like described by Hightower in which HSP70 is exported by the non-classical secretory pathway in the absence of cell death. So the protein have a way to go outside cells without going to the traditional Golgi pathway. So what is this active mechanism for the active transport of HSP70? That's the main question, because this protein doesn't contain a secretory signal that allows the protein to be exported via the Golgi system. So it's exported by this alternative mechanism, the non-classical secretory pathway. And for the immunologists in the room that always use IL-1, IL-1 is the typical protein that go to this pathway. IL-1 doesn't go through the ER Golgi, is secreted by a mechanism that is still unclear in what the protein translocate from the cytosol to outside the cells. And there are many other examples that actually are increasing with time. So how the protein is exported? Well, it's basically two major mechanisms. One is released in extracellular vesicles or exosomes, or is released by another mechanism, though the endolysosomal dependent pathway, that it will be an alternative to a secretory pathway regulated by vesicles in what the protein is translocated to these small vesicles that finish fusing with the membrane and releasing the material. However, we are more interested in the extracellular vesicles or exosomes. And the reason for that is that these proteins have been found that is present on the plasma membrane of some cells. So the question is, what is extracellular vesicles of exosomes? They are lipid membrane enclosed small vesicles, in general 100 nanometers, containing typical cellular components such as proteins, nucleic acid, and metabolites. And they are actively exported from all cell types. So how they are exported? Well, there are three classic mechanisms. One is they are exported to this process called, do you see the pointer? Okay.
Yes.
So there is this process that is called multi-vesicular bodies, and the process is relatively very interesting. The protein is on in the membranes. The membrane gets invaginated, go into these vesicles, and these vesicles form vesicles inside vesicles, and it's called multi-vesicular bodies. Later on, these vesicles inside vesicles fuse back to the plasma membrane and release these vesicles outside the cell. What is critical of this process is that these vesicles or these exosomes that are released outside the cells contain the same topology of the plasma membrane. So proteins that were present on the membrane, in the plasma membrane, looking outside the cells, are also looking outside the exosome. So they have the same topology of the plasma membrane. And that's what this process of making these vesicles inside vesicle is so critical to produce these vesicles. But also, the protein, these vesicles can be shed through the plasma membrane in case that in a condition in what, again, the topology of the plasma membrane is exposed on the surface of these vesicles. And these vesicles come in two flavors. There are very large vesicles that is called microparticles, and there are small vesicles that are called ectosomes, to differentiate them from the process of multi-vesicular bodies of exosomes. Unfortunately, the ectosomes and exosomes have the same size, have almost the same density. So it became really very difficult to separate them. So we decided that rather than try to be thinking if the protein comes from an ectosome or exosome, that is a very intellectual problem. We decided that that's too complicated for the current system, and we decided that we're going to call both of them exosomes, just to say a vesicle of 100 nanometers is by definition now an exosome. There is a third group of particles that is called apoptotic bodies, and these are formed during the process of apoptosis while the cells shrink and they start releasing vesicles. These vesicles are of random size, and there is no regulation mechanism, but they are floating around. So when you try to isolate extracellular vesicles, you're going to have a combination of exosomes, microparticles, and apoptotic bodies. But because the apoptotic bodies tend to be larger, they can be easily separated from the exosomes. Now, what is these exosomes doing? So the idea that we have is that exosomes act as a sentinel signals that prime other cells during the stress conditions to activate a rapid response to avoid the propagation of the insult. So the idea is that cells release these vesicles, and these vesicles can travel. And let me see, I have a slide here. So the idea is that cell, in a normal cell, is releasing these vesicles. I have a very particular composition that they go to the immune system. Here is described as a macrophage, and tell this macrophage everything is okay, there is no action needed. But during stress conditions, the composition of these vesicles change and now go to the immune system and say, oh, there is a stress, and the immune system can preactivate a response to avoid the propagation of the initial insult. So that's why we say that these vesicles are sentinels, and the main function of these vesicles containing this exosome is a signaling molecules. So exosomes deliver these components, the ones in the membrane or in the lumen, to target cells that likely result in alteration of gene expression. So when the cell obtains these exosomes, now have signals, and the cells are responding differently. And depending of the content of the exosome, what is going to be the cellular response. Let me show you an example. These are extracellular vesicles isolated from a hepatocyte cell line that was heat shocked. I was incubated with macrophages. Here we have, just medium as a control, and here we have the response. This is TNF-alpha levels inducing the macrophages. Here we have vesicles that were isolated from the same cells but on the non-heat shock conditions. Instead, you use the vesicle from heat shock cells, you have a dramatic increase in the expression of TNF-alpha. And these, and for comparison, we know that these vesicles contain HSP70, and we calculate, we did an experiment where instead to use HSP70 associated with vesicles, we use the same concentration in the preparation of HSP70, but now in solution. And the difference between the two is the same amount of HSP70, except that here is in vesicles and here is not. The difference is a 2/250-fold difference. Let me show you another example. This is done in culture, so you will say, well, maybe that's an artifact of the culture system. This is another example here. It's again TNF-alpha, different cytokines, TNF-alpha, IL-6, IL-10. These are extracellular vesicles isolated from fibroblasts, from my fibroblasts, and were injected in the peritoneal cavity of macrophages in vivo. And then we isolate the peritoneal macrophages, and by RT-PCR, we quantitate the cytokines. And you can see clearly that if you do, there is a substantial increase in cytokine by these vesicles that were injected in vivo. And that's important for the final of the talk. So now, what are the characteristics of these exosomal extracellular vesicles? Well, the main problem with them is that they are heterogeneous, even when they are released from a single cell source. So there is no this concept is a homogeneous population of vesicles released from cells. No, every cell is releasing a bunch of vesicles, and almost every vesicle is going to be different than another vesicle. The compositions are not identical. And the reason that the composition is not identical is because the content of macromolecules in a small size vesicle of approximately 100 nanometers is very small. So we have calculated that in a single vesicle of 100 nanometers contain only about 200 polypeptide proteins copies. No different proteins, not 200 different proteins, 2 different polypeptides, not different polypeptides, 200 polypeptides. So you can imagine that if the cell produces, it's very strange that we'll have 200 of the same polypeptide. It's going to be a combination of them. And people are very much interested in microRNAs in the exosomes, and there is technical reasons for this interest. But when you calculate how much microRNAs are in one vesicle, you find that there is less than 1 copy of microRNA per vesicle. So not all the vesicles contain microRNA. So when you have this population, what we're trying to say here is that you really indeed are dealing with a very heterogeneous population of vesicles. So the question is, if these are so heterogeneous, how they activate cells. And there is two potential mechanisms. One is that a cell captures a very large number of vesicles, and these vesicles act in a synergistic manner to activate a specific response. The alternative is that maybe these all vesicles are capturing one cell contain a common feature. For example, a common polypeptide that activate a response. Now, this is an example here to show you how much extracellular vesicles can take. So we took extracellular vesicles from cells, and we biotinylate the vesicles and incubate them with macrophages. And here we have macrophages. This is non-permeabilized cells, so this is just binding. And you can see here that there is some red dots that are the exosomes binding to the macrophages. But when you, that obviously detected with extractoavidin. But when you permeabilize these macrophages and detect the biotin within the exosomes, you can see that the massive amount of vesicles inside the cell is huge. So it makes a lot of sense that the cells are taking a lot of vesicles, and the vesicles, because there is some chance that it's going to be some kind of common component, can activate the cells. However, so basically here what we are saying is that there are two mechanisms, synergy versus a common component. Common feature of all vesicles is the lipid membrane. So all of them, in spite of what proteins or microRNA they contain a lipid bilayer that is formed mainly by phospholipids and cholesterol. So the question is, can these phospholipids that are the main constituent of this membrane of this exosome can play a role in the activation of cells? And a simple way to investigate what is the role of phospholipids in cell activation is to study the effect of specific phospholipids in liposomes. Liposomes are just vesicles made of a lipid, could be a combination of phospholipids or one phospholipid. Remember, the phospholipids have a head that is polar and a tail, so when you put phospholipids in water, they tend to form a vesicle that you can size of any size that you want. So, and the interesting thing is that you can make liposome or a specific phospholipid or with a combination of phospholipids. So for this study, what we did is that we prepare liposomes of two kinds of lipids. One is PC, that is phosphatidylcholine, or from phosphatidylserine. Now, what is interesting about this? The main component of cellular membrane is phosphatidylcholine. And obviously that should be present, is the main component in the vesicles. However, in the plasma membrane, the inner part of the membrane contains phosphatidylserine that is not present on the surface of the cells, except in apoptotic cells. So if you take a piece of membrane, contain PC outside the cells and PS inside. However, these lipids tend to flip, and this asymmetry of the plasma membrane is maintained by a process that is regulated by specific enzymes using energy to maintain PS inside and PC outside. But when you have these two lipids in an exosome, the machinery to keep the PS inside the cell is not there, and consistently, these exosomes now are going to have PC and PS on the surface of the liposome. So we have these two liposomes, and you notice that we use palmitoyl or allele in both cases because this is the same fatty acid. So the difference between these two is just in the head. PC is neutral and PS is negative charge. So we took these liposomes and we inject them into mice in the peritoneum. We inject about 10 to the 5 liposomes to macrophages. and we use PBS as a baseline. And then we harvest, after 1 hour of the injection, we harvest the peritoneal macrophages. And what we found is that these peritoneal macrophages, let's go to the next slide, are totally captured by macrophages. I don't know if you see the markers. My screen is very small, but here is F480. That is the marker of macrophages. And here is the density of it. We use liposomes that were labeled, and the liposomes were captured. And actually, when you calculate, all the liposomes are captured. These macrophages are capturing all the 10 to the 5 liposomes that we put in the cells. And there is some kind of quantification here. I cannot see well that slide because it's too small. And it's also internalized. Here, cells incubated with these fluorescent liposomes, and you can see that with time, you see more liposomes inside the cells. And these are don't need permeabilization because the liposomes are charged with a very strong dye. Interesting enough, these liposomes are not internalized by the traditional receptor-mediated process, are internalized by pinocytosis. And the reason that we say that is because you see that there is no colocalization with transferrin and the transferrin receptor. That is a typical endocytic pathway, the clathrin-dependent endocytic pathway. They go for a different pathway like this, and we believe that is micropinocytosis. Now, the interesting thing is that we took these macrophages that were incubated with these liposomes, and now and we isolate the cells, and we isolate RNA and do RNA-seq of these cells that were incubated with PS or PC liposomes. And what we found was that PS modulated the expression of 4,000 genes in these macrophages, whereas PC only activate the genes of about 1,600 by PC. All of this is in comparison with PBS, and these numbers, 4,000 and 1,000, is respect to 12,000 genes that were detected by the RNA-seq method. So about this liposome is activating about 37% of the, it's responsible for the activation of 37% of the RNA, of the genes in these macrophages. Here is just on the data. What is important is in the next slide. We compare now the genes that were specifically activated by PS versus PC, or PC versus PS. And what we found is that there is different genes. There are 14,000 genes that were specifically altered by PS, but not by PC. And 361 genes that were modulated by PC and no PS. So now we have a separation of genes that were stimulated by each one of these lipids. And remember that we are using the same fatty acids. So these only depend on the head of the lipid. And we do the typical signal analysis pathway. We found different pathways, but the interesting pathway here is this, the cytokine receptor pathway. And when you look into this, and this is a very, most of the significant pathway was the cytokine-kinokine receptor interaction, in which 27 genes, okay, in that pathway were significantly regulated. For those that have done RNA-seq, when you define a pathway, sometimes you have 2 or 3 genes that are alter in this pathway. You see this pathway is significant. In this case, we have 27 genes that were specifically upregulated by exposure to PS liposomes. And this is a very busy slide. This is all Here it is. That's a very busy slide. All of the genes in the pathway, and here we label the ones that were altered. And 27 is a very large number. What is important in this case is not just that you find in the pathway, you need to validate them by different methods, and we use RT-PCR. So now we took cells, exposure up to the two different lipids, and independently of the RNA-seq, we did RT-PCR for each one of the genes that we thought that we were regulating. And you can see that the cytokines, and unfortunately I cannot see well the names of them. Maybe I can do this. Here I have it. So this is TNF-alpha. Never mind.
Yeah, we can see our pointer
different cytokines and chemokines. Different cytokines and chemokines were all specifically induced or expressed by PS and OPC. And to demonstrate the robust of our analysis, here we have IL-10. And IL-10 is equally regulated by PC or PS. So this is a highly specific effect of PS liposomes with respect to PC liposomes. Now, here in the top are chemokines. And the function of chemokine is to attract neutrophils to the site in what they are released. So if you see an increase of chemokines, will you think or speculate that if you inject PS liposome in the peritoneal cavity of a mouse, it should be an increase in the recruitment of neutrophil to this cavity? And so what we did is the same experiment. We do the liposomes and we see if there are neutrophils in there. This is the flow cytometer data. Let me show you this here. It is very clear. There was a massive infiltration of neutrophils in the peritoneum. A massive infiltration of neutrophil in the peritoneum that was observed after PS, but not after PBS. And the same thing with PC. And that data can be quantitated. And we have information that if you wait now, say these macrophages stay in the peritoneal cavity for a few days, but after 17 days or 14 days, they disappear. Okay. Oops. I see what happened now. Okay. So if you have peritoneal neutrophils in the peritoneum, you will speculate that if you have a localized infection, they will take care of the infection very quickly because they are already there. So what we did was a model of sepsis in what is a model of multi-organism invasion. And basically what you do in this model is that you make a hole in the cecum, that is in the gut of the animal, and that release all these microbes that are in the lumen of the gut inside the peritoneum and create a major infection and inflammatory response. Now, what we did in this experiment is that we inject the animals with the liposomes. I wait until the neutrophil were infiltrated and did a cecal ligation and puncture. And here we compare the data between animals that were injected with the PS liposomes and animals that were injected with just PBS. And you can see that there is tremendous protection in this insult if you have PS liposomes, but you don't see it if you inject PC liposomes because they don't attract neutrophils to the cavity. And interesting enough, for those that like to compare genders, females and males have exactly the same effect. So I'm gonna switch gears a little bit about these exosomes, and I will go and say that pathological conditions are likely to result in changes of the exosome or extracellular vesicle composition. Therefore, they may be used as disease diagnostic of a biomarker. And you know, in the typical situation, let's say this cartoon, if I have a better idea what's wrong with you when I receive the result of your autopsy, test, the result of your test. So we don't have to wait for the autopsy to know if we have a good marker. Now, extracellular vesicles are being detected in a large number of diseases, and here is many of them, and this number is growing, and I cannot keep up with how many more diseases appear every day, but they are present. And remember, these extracellular vesicles are in the body fluids. Now, extracellular vesicles can be isolated from a variety of body fluids, such as blood, saliva, and urine. And there are different methods to do that. Now, urine is an ideal body fluid for the study of extracellular vesicles during disease conditions because it can be obtained in great quantities, and more importantly, the acquisition is painless. So you don't have to use fine needles. You just need to find a person that is just gonna dispose the exosomes in a container and give it to you. And I always say that by the time that I finish this talk and you go to the corridor and you go to the restroom to release yourself, think about how many exosomes are you dumping into the sink. So we established a protocol for isolation of the urine exosomes, and the key factor is that we need to add zinc sulfate, and I'm not going to discuss why, but it works. It helps in the storage of the exosomes, can be stored at -4 degrees for 7 days, or -20 or -80. The process is differential centrifugation. You go a low spin in what you pellet cells and cell debris. Later on, it's a higher speed centrifugation where you pellet larger vesicles like the apoptotic bodies or the microparticles, and you do a final high speed centrifugation where you get the exosomes, and these you determine the size and the composition. There is many ways to do that. I don't know if I have a picture here. Okay, what is important here is that what we evaluate is could exosome from urine be used as a potential biomarker? So we don't have a disease. This is a healthy person. And what we did is that we took the same person and we collect urine 2 times a day during different days. And we quantitate how many exosomes were in the urine per ml of this person. And remarkably enough, it doesn't matter which day or what time of the day, you always have the same number of exosomes. It's about 1 per 10 to the 10 exosomes per ml. That's remarkable because that means that whenever you are urinating, you're producing the same number of exosomes. That's what I said. Remember when you go to the restroom, how many you are wasting there. But we also say, well, maybe what is the variation between people? And this is 5 different subjects. And we isolate the exosome from them. And again, the numbers are very similar. There is no tenfold difference between one individual and another. And by the way, some of these are females and some are male. Okay, so what we did is that we took these urine exosomes and we do a characterization of the proteins that were in the exosomes. We decide that if you take this exosome and you do mass spectrum, you find thousands of different polypeptides. So we say, well, we have to be more restricted than that. So we decide to focus on membrane proteins. And the majority of membrane proteins in the exosomes are glycoproteins. So what we did is that we did a pull down of using CONA, that is a lectin-binding glycoproteins, and we pull down the glycoprotein for these exosomes at the mass spec and find what is the most relevant proteins in there. And there are 4 here that you can see: CD26, DP1, BSP4, and GAL3. And these are Western blots of urine sample, and you can see that the Westerns are beautiful. They have plenty of these proteins. So these are normal people, and the question is, well, what is the variation of these proteins between individuals? And here we have males and females, and you can see that some of them are totally constant in the urine preparation of some individual. Some of them have some changes. Okay, so maybe there is some kind of variation. I don't think it's significant because it's not consistent, but still, you know, it's amazing that most of the people contain the same number of the glycoproteins in these exosomes. So let me summarize the talk, saying that extracellular vesicles of exosomes mediate intracellular communication. The activation of cells by extracellular vesicles of exosomes is very complex, in which all their components may participate in changing biological process. Phospholipids with the exosome have a great impact on macrophage gene expression. Exosomes or extracellular vesicles are a potential marker of disease, and extracellular vesicles of exosome isolated from urine could be a prospective diagnostic. And with that, I'm going to finish. This is a picture of my lab. That's the time that Christina was visiting us. This is our favorite restaurant in San Diego, Berta's, where we always have the Christmas lunch. And you can see I'm a raving fan. And this is Paulo, that is also from Brazil. This is the people that was involved in this project. All of these in red: Debbie Cowitt, James Prieto, Paulo, Virginia Vega, many people that was involved in this project. And with that, I would like to take any questions.
Very cool, my friends. I love it. This is the San Diego sunset, the famous.
That's the San Diego sunset.
Exactly. Fantastic. Thank you so much. That was a great talk. I'm gonna give people some time to type up the questions here in the chat, but I already have some for you because, you know, I'm a fan of the lipid vesicles model. So I don't know if people, everybody could follow, but it's very interesting how the two talks, the one we had before by Info and now the one by Antonio, we take very complex problems. One is the immunological synapse, and here is the extracellular vesicle. And they take a reductionist approach and they go to the core. It's very hard to work with extracellular vesicles in bulk, right? Because yes, it's a black box. We don't know what's going on in there. There's RNA. Now you did a really nice job quantifying the amount of RNA. You can see, well, not all the vesicles have RNA in them, right? And there's protein, there's a lot of other stuff. But the way you did it, when you simplified it and you said, well, let's redo the whole system. Let's start from scratch. And let's take the POPS and the POPC liposomes and see if this is something. There's something different. And then
let me tell you something. Let me tell you something that is very important I forgot to mention is, look at the effect of these pure lipids, pure phospholipids on the immune system. Now, what is this important today? The Pfizer and Moderna vaccines
are
made of mRNA, make liposomes.
Yeah.
So the idea is that the effect of these vaccines is not just because the mRNA that they contain, is that the lipids can act as adjuvants and enhance the response. And that's something that nobody talks.
That's true.
So
nobody says anything about it.
Probably, well, because nobody work with lipids. That's true. Immunologists don't like lipids. So
that's true. No, they don't know anything about it. Exactly.
And I think a key factor in the success of that vaccine is the fact that the mRNA is encapsulated in a lipid environment.
And that's what I was going to, because when we think about how, just a different lipid in the composition of the vesicle can change gene transcription, how the lipid can actually affect a very important axis for immunology, which is the cytokine receptor axis. So do you know what's the lipid composition of these vesicles that they're using Do we know anything about it?
No,
It's totally
that's a big
proprietary.
secret. It's
Yeah.
a totally secret. I
Yeah.
will guess that they are made of PC. Most of the people that work with liposomes use PC, a little bit of cholesterol, and there is something else that they add to. They put something else to stabilize the liposomes, but it's mainly PC. I'm pretty sure about that. And PC also have an effect. What happens is when you compare PC versus PS, the effect of PS is so large that is enormous, correct? But PC have an effect.
And PS has this receptors, right? But what you're thinking is that it doesn't matter, that it's just going to go through the membrane and it's going to change the charge of the membrane, right?
I eliminate that slide because I didn't want to overwhelm people with this. We know that it's not a receptor-mediated process, the uptake of these liposomes. It's not the PS receptor. And I will tell you why. We use liposomes made of phospholipids D and L, the two stereoisomers.
The isomers.
And, receptors are stereospecific. So the fact that the two, L and D, have exactly the same effect demonstrates that this is not a receptor-mediated process. It's due to the internalization of the vesicles inside the cells. Now, what happened with these vesicles when they go inside the cells? I don't know. That's what we're trying to investigate.
And
there is another problem there. So now when we're dealing with the lipids, there is two components. The lipids still have the head. So when you talk about difference between PC and PS, the head is what could be important, but also the fatty acid can have an effect. So some of the genes that can regulate, like IL-10, that could be regulated by the two phospholipids, could be due that is the fatty acid will regulate the expression of IL-10.
Exactly. This is a new concept. I think this is very interesting. And I think this is going to become of paramount importance now because of the mRNA vaccines. I think you're totally right. Everybody's thinking about the TLR receptors that mRNA is going to activate and how it's going to activate the cells, but they're not talking about how the lips are going to do it. So I think this is a major field for you too. It's very interesting. So I have a question here in the chat, two questions. One is from Thiago. He says, "Hi, Antonio, great talk as always." And he asks, do these exosomes always are they always internalized, or are they incorporated in the membrane of the acceptor cell? Like cross-dressing. I don't know if you're familiar with cross-dressing in immunology.
Yeah, that's an interesting question. We don't know. The exosomes could be interacting with cells by a receptor-mediated process. They can be internalized by endocytosis or pinocytosis, but also they can fuse with the plasma membrane and incorporate the cargo. So if you think a liposome fused with the plasma membrane, the membrane protein of the exosome will remain on the plasma membrane, and then maybe later they are going to be internalized. But the cargo, the one is in the lumen of the liposome, will be directly into the cytosol. We don't know that. And I believe that for the exosomes, all of them are going to be valid.
Yeah, exactly. I think these are actually testable hypotheses because you can test all of them. There's another question here. What is the main challenge to use the ECV in diagnostic? Because we were talking about urine.
The main challenge is to define a marker that is going to be specific for a disease. Isolating the vesicles is not a problem. Urine is great. Blood is a little bit more challenging because they tend to be contaminated by all the coagulation factors. But you can have it in saliva, depending on the source. The challenge is that you find a specific marker that is related to a disease. You find that you are in a gold mine. That's why people are paying attention to microRNAs, because microRNAs are easily to detect, and because there are less, there is more chance that you can find something related. But the big challenge is to compare So if you take exosome for a healthy person and a person with a disease, and you do mass spec, you find million things, and it's very difficult to discern what is what. So that will require a little bit more work to define what is the marker.
I remember 10 years ago, I was at a meeting, a nature meeting, and some guy, I can't remember his name right now, but it was like this big shot in the field, and he was saying, you know, next month we're going to be able to diagnose any tumor based on the extracellular vesicles to see if it's a metastatic or if it's a primary tumor. And this was 10 years ago, so I guess it did not work. It's very complex.
The other thing that they are using exosome is a cargo delivery. So they are putting drugs in the exosome. Rather than putting the drugs in the liposome, they are putting the exosomes. And I tell you, maybe some of these vaccines that are done with liposomes maybe could be even more effective if you put the mRNA into that exosome.
Yeah, but then if you add the RNA in the exosome, then I mean you can control. I like your system because you can control the lipids.
Oh, absolutely.
And the other thing that you didn't talk about, but we talked about when I was in your lab, is that every different cell type has a different composition of specific lipids, and that is reflected on the exosomes, the extracellular vesicles that they release. And do you know if this can be altered by stress? Do we know this already, or by activation state?
Well, if you stress cells, the lipid composition of the plasma membrane change. And in cancer cells, and that's Gabriel Multov's work, show that in cancer cells, there is some kind of lipids that are more abundant in the cancer cell versus the normal cells. So the lipid composition changed a lot. What happened is that, again, nobody wants to work with lipids.
Yeah, no, but I think this is going to change. I'm pretty sure. So I just wanted to stress out that we know, people from the graduate program know this already because we talked about it. Dr. De Mayo is going to be available in the next weeks. We can find a good time. So when professors and students can get together for a Zoom talk, and we're going to talk in person and discuss maybe projects. I was thinking maybe we can set up like a course, you know, online before we can travel, and then afterwards, you know, we are going to be so happy when you can finally visit us and do this in person. You want to put me
to work, huh?
Yes, absolutely.
Well,
but then it's going to be nice because then you can come here and drink caipirinha and we can talk about more science.
That's better. After the holidays, we do that.
Absolutely, absolutely. Thank you so much, Antonio. That was a wonderful talk. It was great to have you, and I'm sure I'm just getting all the...
Ellie did a great job. Everything worked perfect.
Yeah, it was tough the first talk. I'm so glad everything worked out for you.
That's why I didn't want to do the first. You see, I didn't want to do the first.
He always knows this.
You know, remember the phrase that said the devil knows more for all than for devils. So
exactly.
No, you never go first. You never go first in this kind of things. That's a problem. You let people experiment first.
Yeah, that's true. That's true. Well, thank you so much. Thank you so much for all the questions, and people loved it. I can see this here. So we'll talk later. Thank you so much.
Thank you so much. Good night.
Night.
Thank you, Antonio. Thank you, thank you. Thank you, Cristina. Obrigada, Cris, pela tua excelente condução. Sensacional.
Deu tudo certo. Deu tudo certo no final.
In the end, everything is gonna be alright. So, então, pessoal, eu acho que a gente vai finalizando a nossa transmissão por aqui, né? A gente agradece demais a participação de vocês. Nós tivemos mais que 100 pessoas acompanhando a nossa primeira noite de palestras. E, apesar dos percalços, estamos todos aprendendo, né? Esse novo jeito de trocar ideias, de fazer meetings, enfim. Então, o mais importante é que, no final, tudo deu muito certo. E esses dois colaboradores teus, né, Cris, eles fizeram duas palestras sensacionais, uma melhor que a outra, assim, então a gente agradece por poder contar com a tua assistência sempre, a tua participação, é sempre muito importante.
Imagina, acho que foi legal, que bom que no fim deu tudo certo.
Sim. Então, pessoal, a gente deseja uma excelente noite para todos e para todas. E aí, amanhã a gente está aguardando vocês, às 6h30, com a próxima palestra do professor Josevigny, lá da Universidade Laval, e depois, na sequência, o Dr. Tiago Borges, do MD8, que estava nos acompanhando essa noite também, estava nos abrilhantando. Então, um grande beijo, pessoal. Aguardo vocês amanhã. Ajudem a divulgar o evento com os colegas de vocês.
Tá bom. Até amanhã, então.